Good afternoon to all the EAPCET aspirants. So, here we are dealing with histology and anatomy of flowering plants, where we have already completed with the introduction and how to deal with this chapter, and so far we have completed tissues of this chapter, completely exercised meristematic and permanent tissues in our previous class. So, now in today's session we are going to start with tissue systems beta. First we are going to start with what? Tissue systems. Here if you see, we have 3 tissue systems in plant anatomy, 3 tissue systems. Take the notes parallelly ma. 3 tissue systems. Which includes epidermal tissue system, ground or fundamental tissue system, and vascular tissue system. So, here today we will learn all the 3 tissue systems along with the comparative analysis between dicot monocot root, dicot monocot stem and dicot monocot leaf. So, here you see first epidermal tissue system. Epidermal tissue system includes only epidermis. Okay. It only includes what beta? Epidermis, which is a single layered parenchymatous layer. Okay. Single layered made up of parenchymatous cells which are elongated with less intercellular spaces in between. Okay. Epidermis is single layered in most of the plants. In few plants like Nerium, you can see multi-layered epidermis. Multi-layered epidermis. So, how can you describe epidermis as a single layered? It is unilayered, made up of parenchymatous cells, and it has elongated cells like this. Okay. So, and what does this epidermis consist of, or epidermal system consists of? Along with epidermis, they possess epidermal appendages. Epidermal appendages. Epidermal appendages includes stomatal apparatus in the leaf, trichomes in the stem and root hairs in the root. Okay, root epidermis. So, this is a brief description of epidermal tissue system. As of now, you have to be clear like what are the layers that are included under epidermal tissue system, that is only epidermis. Clear, right beta? Next is ground tissue system or fundamental tissue system. We will come to this after looking into vascular tissue system. Vascular tissue system includes the complete vascular bundles. Okay. Vascular tissue system includes vascular bundles. Only vascular bundles, which includes xylem and phloem. Fine. This vascular bundle arrangement will be different, like conjoint, collateral, radial, bicollateral in root, stem, leaf of different plants. We will look into different types of vascular bundles in the coming slide. As of now, you remember like what are included under epidermal tissue system beta? Epidermis is included under epidermal tissue system. Under vascular tissue system, what is included? Vascular bundles are included in vascular tissue system. Apart from epidermis and vascular bundles, all the layers in the plant anatomy or tissue systems are included under ground or fundamental tissue systems, which includes below the epidermis, what is the layer that is present beta? Below the epidermis, that is hypodermis, which is present. Okay. Ground tissue system includes hypodermis, after hypodermis it is general cortex. Okay. It includes general cortex. General cortex consists of what? General cortex, if it is in shoot, general cortex consists of hypodermis, general cortex layer, that is cortex layer and endodermis. So, here you can take below the epidermis, that is cortex region, which includes hypodermis, general cortex, and endodermis. Fine, ma'am. So, this include hypodermis, general cortex and endodermis in cortex region. After cortex, stellar region comes into picture, right? Stellar region. Stele includes pericycle, vascular bundles, though it comes under vascular tissue system. Vascular bundles, it comes over here, okay, and it also includes pith. Pith or medulla and medullary rays. Fine, right. So, here we have epidermal tissue system, ground or fundamental tissue system and vascular tissue system. The very beginning that you have to know is, in epidermal tissue system, it consists only epidermis and epidermal appendages. Epidermal appendages, if it is on leaf, it includes stomata or stomatal apparatus. If it is on the root, so unicellular root hairs. If it is on the stem, so multicellular hair like projections, that is trichomes. After which vascular tissue system, which includes only vascular bundles. In our next slide we will learn what are the types of vascular bundles, and in ground or fundamental tissue system, it includes cortex layers. Cortex layers are what, ma, here? Hypodermis, general cortex and endodermis. Endodermis is the last layer of the cortex, and after cortex region, stellar region comes into picture. In stellar region, which is at the center, it includes pericycle, vascular bundles and pith or medulla and medullary rays, of which vascular bundles are under vascular tissue system, and it includes pericycle, pith and medulla under ground tissue system. So, here you have to remember that apart from epidermis and vascular bundle, everything or every region or layer is included under ground or fundamental tissue system. Now, we will look into different types of vascular bundles. Okay. What are different types of vascular bundles? Look at here, different types of vascular bundles. We have two types of vascular bundle arrangement. One is open form of vascular bundles and second is closed form of vascular bundles. Closed form and open form. What is closed form of vascular bundles? If there is no cambial region that is present in between xylem and phloem, it is considered as closed form of vascular bundles. Okay? No cambium, no cambial layer between xylem and phloem. Okay, between xylem and phloem, there is no cambial layer. If at all a cambial layer is present, a cambial layer is present in between xylem and phloem, it is said to be open form of vascular cambium. And next, based on the arrangement of xylem and phloem tissues inside the stellar region, we have different types of vascular bundles. Okay? Types based on whether cambium is present or not, and types based on the positions of xylem and phloem, which includes radial. Okay, it includes radial, conjoint, collateral, bicollateral, okay, these are different types of vascular bundle arrangements. So, here first is radial type of vascular bundle arrangement. Here in the radial type, if you see ma, xylem and phloem are at the different radius. This you will come across in the root, okay, root, wherein xylem and phloem are at the different radii, side to side, okay. Xylem and phloem are at the different radii, that is called as radial type of vascular bundle arrangement. Here you can take down xylem and phloem at different radii. And here you see conjoint type of vascular bundle arrangement, wherein xylem and phloem are at the same radius, okay. Xylem and phloem are at the same radius, that is conjoint type of vascular bundles, okay, wherein xylem and phloem are at the same radius. Fine. Now, collateral. If xylem and phloem are side to side of each other, they are side by side of each other, then you will address that type of vascular bundle as what? Collateral, wherein xylem and phloem are side by side of each other. Bicollateral, this is mostly observed in gourds, wherein xylem is sandwiched in between two phloem tissues like this, okay. Xylem is at the center, which is sandwiched by two phloem tissues at the either ends, that is called as bicollateral type of vascular bundle, okay. Xylem is sandwiched by phloem at two ends. These are different types of vascular bundles. One is based on presence or absence of the cambium. If there is no cambium, you will call it as closed type. If cambium is present, it is open type. Then based on the arrangement of xylem and phloem, if xylem and phloem are at the different radii, that is called as radial type of vascular bundles arrangement. Next, conjoint, if xylem and phloem are at the same radius, and collateral, xylem and phloem are side by side to each other. Bicollateral, which is mostly observed in gourds like pumpkin, cucurbitous and all, wherein xylem is sandwiched by two phloem tissues. Xylem at the center, sandwiched by two phloem tissues. These are different types of vascular bundles. Now, dicot and monocot root comparative analysis with three tissue systems we will see with. After dicot monocot root, we will move on to dicot monocot stem and then dicot monocot leaf. First you see dicot monocot root. In dicot monocot root, if you observe keenly, just a second. Yeah, I hope now the images are clear to you. Yeah. So, look at here ma. First, dicot monocot root, first is what? Epidermal tissue system. Epidermal tissue system. Epidermal tissue system here includes what? Epidermis. Here epidermis is also called as epiblemma or piliferous layer. Okay, this is also addressed as epiblemma or piliferous layer or rhizodermis. Fine. This is called as epiblemma because it absorbs water from the soil. Piliferous layer because it has unicellular root like projections, unicellular hair like projections called as root hairs. Okay. Root hairs here are what? Unicellular projections, unicellular tubular projections. Fine, right ma. So, here that is called as piliferous layer and it is also called as rhizodermis because it has root hairs over epidermis. Remember the synonyms properly because these synonyms are used only for root epidermis. Clear, right beta? Clear with root epidermis? Fine, right. So, now in the cortex region, unlike that differentiation that you see in stem as hypodermis, general cortex and endodermis, here in the root cortex region, there is no hypodermis. You just have what? General cortex. Okay, you just have general cortex and endodermis. Fine, right. General cortex, which is made up of parenchymatous layer again, and then endodermis. Here there is a peculiar feature that you have to mark on the endodermis of the root, where many a times asked question that root endodermal cells will have suberin depositions in the radial and tangential walls. Okay, they have suberin depositions in the radial and tangential walls like this. This suberin depositions will prevent the apoplastic movement, and symplastic movement of water is promoted from the endodermis. I will give you a brief description. First listen that at the endodermis region what is present, ma? Suberin depositions in the radial and tangential walls of root epidermis. What is present? Yeah, suberin depositions. Suberin depositions. So, in transport of plants and plant physiology, if you observe, where we have learnt that when the absorption of water has taken place from the root hair region till here in the endodermis, the water is passed from the root hairs through two types of movements. Those movement is what? Apoplastic movement and symplastic movement. Okay. Those ways are what? Apoplastic and symplastic movement of water. Apoplastic movement means water will travel across the cell walls in between two different cells. Okay. Water travels in between cell walls of two different cells like this. Okay. But symplastic movement is water travels through the cell's cytoplasm. Water travels through the cell's cytoplasm through plasmodesmatal connections. Okay. Water travels through the cytoplasm of the cells, through plasmodesmatal connections. Plasmodesmatal connections. Clear, ma? So, through plasmodesmatal connections, the water is being transported intracellularly, symplastic movement. So, both apoplastic and symplastic movement is being done till the endodermis. In the endodermis, what happens is, as suberin depositions are there in the radial and tangential wall, what happens? Suberin will not permit the flow of water which is in between cells. So, from endodermis, it is strictly symplastic movement to the inner side, to the stellar region. Okay. To the stellar region, what is the movement of water from endodermis? It is strictly symplastic movement. Remember that we can get a question in combination between transport in plants and anatomy of root. Okay. Remember apoplastic and symplastic movement very strictly. Next, in stellar region, if you observe, in stellar region we have pericycle which is parenchymatous again, it is single layered parenchymatous pericycle is present in the stellar region. So, here next is what? Stellar region. In the stellar region, first layer is pericycle. After pericycle, you have xylem and phloem tissues, that is vascular bundles arranged. These vascular bundles are how they are arranged, beta? They are arranged in radial fashion, that is xylem and phloem are at different radii both in dicot root and monocot root. Okay. Second is vascular bundles. These vascular bundles are radial. Okay. Radial vascular bundles. Fine. Radial closed in monocot and open in dicots. Okay. Radial closed in monocots and open in dicots. Open in dicots. That is, the tissue which is present in between xylem and phloem here is called as conjunctive tissue. The tissue which is present over here is what ma? Conjunctive tissue. This conjunctive tissue will get meristematic and it forms a wavy cambial ring. Okay. This forms a wavy cambial ring like this. Like this, a wavy cambial ring is formed. Fine, right. In between xylem and phloem, from the pericycle region, okay, cambium arises in between the conjunctive tissue attaching the pericycle. Cambium forms a wavy cambium is formed in the dicot root. Okay, wavy cambium. From where this wavy cambium arises, beta? This arises from pericycle. Arises from pericycle. Fine, right. So, next after vascular bundles, third is what? Third is pith or medulla. Pith or medulla. Fine. There is one simple differentiation between dicot and monocot root here. In dicot root, the number of vascular bundles can be 2 to 4, diarch to tetrarch. Whereas, in monocot root, the number of vascular bundles can be more, like it can be more than 6. So, you will call it as polyarch vascular bundles. Polyarch vascular bundles. And at the center, pith is small and inconspicuous in dicot root. Pith is small and inconspicuous in dicot root, and pith is large in monocot root. These are the simple differentiations. Everything else is same in between dicot root and monocot root. Epidermis is same. Epidermis is also called as piliferous layer, epiblema or rhizodermis, both in dicot and monocot root. Second is what? Cortex region. Cortex region just has general cortex and endodermis over here. Endodermis has suberin depositions in the radial and tangential walls, because of which only symplastic movement of water is promoted from the endodermis to the stellar region. Okay. Till here it is both apoplastic and symplastic movement of water. After endodermis it is stellar region. Stellar region consists of what? Pericycle. Okay. In morphology we have clearly learnt that lateral roots will arise from the pericycle. Lateral roots endogenously they will arise from pericycle. So, here in monocot root only lateral roots will arise endogenously from the pericycle, but in dicot root both lateral roots and the cambium will arise from the pericycle. Okay, both lateral roots and cambium will arise from the pericycle region. Next is vascular bundles. Vascular bundles are diarch to tetrarch in the dicot root and polyarch in the monocot root. Here you can see small and inconspicuous pith, and here you can see large pith, okay, in monocot root, and closed vascular bundles in the monocot root, here open form of vascular bundles. Clear, right? These are the basic differentiations that you will see only in the stellar region. Now, we will make a clear note on the differences between dicot and monocot root in a tabular fashion, highlighting the tissue systems. Okay? Yeah. So, let us get through ma. First, dicot root and monocot root. Dicot root, monocot root. First, we will go through the epidermal tissue system. Take this table, very important. If you just have this table also, it is enough ma. You can go for a revision directly with these tables. Fine, right. Here it is tissue systems. First, epidermal tissue system. Epidermal tissue system, both in dicot root and monocot root, it has only epidermis, which has elongated cells, unilayered, okay, and this epidermis is also called as rhizodermis, piliferous layer, Okay, it is also addressed as rhizodermis piliferous layer. Fine, right, and epiblema. So here, and this epidermis is unilayered, it has unicellular tubular projections called as root hairs, both in dicot root and monocot root. And next you see ground tissue system. Ground tissue system in dicot root and monocot root. Both has cortex. Cortex is parenchymatous. Okay, and then both have endodermis. Endodermis, the cells have what depositions? Suberin depositions in the radial and tangential walls. This suberin depositions in the radial and tangential walls will prevent what water movement? Apoplastic water movement. So, this suberin depositions will prevent apoplastic water movement, and it will promote symplastic movement of water through what beta? Through plasmodesmatal connections. Through plasmodesmatal connections. Clear, right? Next, third is after endodermis, yeah, in the general cortex, pith is also included, like under stellar region. Next is pericycle. Pericycle is again parenchymatous in both of them. There is one differentiation, that from pericycle here, from pericycle in dicot root, both lateral roots and cambium arises. Here only lateral roots arise in the monocot root. Lateral roots arises. Next is fine, right? Yeah, so here cortex, endodermis, pericycle differentiation I have highlighted. After pericycle, under ground tissue system, pith is included, pith or medulla. Pith or medulla is again parenchymatous. Fine, right? And then we are going to learn the fundamental tissue system. Yeah, fundamental tissue system. Sorry, vascular tissue system. Third is vascular tissue system. So, this vascular tissue system includes only vascular bundles, we know that, but here what type of vascular bundles are present in the dicot root? We have learned, right? Radial and open type of vascular bundles. And how the open type of vascular bundles is formed? Wavy cambium is formed. Here you can see that wavy cambium, like xylem at the center and phloem, then xylem and phloem. A cambium is formed attaching to the pericycle like this. Okay, this is the wavy cambium that is present. Fine. So, open radial and open type of vascular bundles, and it is diarch to tetrarch, that is 2 to 4 vascular bundles will be present, and pith is small and inconspicuous. Small pith is present. Here if you see in the monocot root, how many vascular bundles are present beta? Vascular bundles are radial and closed, radial and closed, and here you can see polyarc type of vascular bundles, polyarc type of vascular bundles, and pith is large, okay, large pith is present. So, this is the complete description of epidermal tissue system, ground tissue system and vascular tissue system of dicot and monocot root. You can go through that clearly and take a screenshot. So, yes, stem and leaf, comparative analysis between dicot stem, dicot leaf, dicot stem and monocot stem, dicot leaf and monocot leaf, we will do it in the next class. Till here you make a clear note of it beta, tissue systems, dicot, monocot root, clear analysis. If you have this single slide also, it is enough. Clear, right? Yeah, that is all for today's session. Thank you.